So we know that glucose is not the only thing that's made. Oxygen gas is also produced, right? So basically, the previous experiment, it was showing us whether glucose was produced, whether starch was present. We looked at the conditions, light and chlorophyll. So now we could look at whether oxygen is produced during photosynthesis, right? So we know that glucose is not the only thing that's made. Oxygen gas is also produced, right? Now, if we were to look in our yards or in our neighbor yard or whatever, we could think that the plant has been photosynthesizing, but it's hard to actually know, right? We can't actually see the glucose being made. So we have ways of testing it to see starch is present. But if we look at the plant, we can't also see the oxygen gas coming out from the stomata, right? So what we could actually do is see if photosynthesis is occurring in water plants. So what happens is, well, this is the method here, but this is basically how it looks, right? So you take a funnel and you put it over some pondweed because that's a plant that photosynthesizes in water, so it's a marine plant. And then we fill a test tube with water and place it upside down over the funnel stem. The reason why we fill in the test tube with water is, again, we want to see the gas bubbles inside of the water, right? Now, we could do this to see if photosynthesis has occurred. So we put it in sunlight and we want to see if any oxygen bubbles are happening inside of this test tube, right? So the idea is that if photosynthesis occurs, one, we'll actually see the oxygen bubbles, right? But two, there is something called a glowing splint. What a glowing splint is, it's another type of indicator. So it's basically this stick, right? And upon contact with oxygen, or if oxygen is present, it will light on fire, right? So what happens is, if there's oxygen present in the test tube and we put the glowing splint in, we expect that the oxygen would allow the glowing splint to catch on fire, right? So this is another way to observe photosynthesis, right? So again, if CSEC asks you to design an experiment to investigate whether oxygen is produced during photosynthesis, this is something else that you could do, right? So the idea again is that once this plant is photosynthesizing, so let's say that we have some sunlight here. So it's giving off sunlight. Inside the plant has chlorophyll. The plant has carbon dioxide and it has water. So it's photosynthesizing, right? So once photosynthesis is occurring, we expect that glucose would be produced and we expect that oxygen would be produced, right? Now remember, again, oxygen is a gas, right? When this plant is photosynthesizing and it produces the oxygen, what you see here is this gas bubble. The reason why it has a gas bubble is because it's producing oxygen after it photosynthesizes. So all these things that you see in here, they're all gas bubbles of oxygen. Now, the reason why we turn the test tube upside down, why we put water in the test tube and turn it upside down, is because you want to visibly see the gas bubbles, but you also want a kind of collection center. So that's why we use a tube. So the oxygen is just not escaping into the atmosphere, right? So after we collect oxygen in that tube, we quickly remove the tube and then we'll put the glowing splint inside of the tube, because once this glowing splint comes into contact with the oxygen, it will ignite, so it'll catch on fire and we'll visibly be able to see the reaction, so we know that oxygen was present in the tube.